Everyone, welcome to my channel Pure Biology, and today I come with another video, that is the embryogenesis of the plant. And in my previous videos I have already explained about the fertilization, plants fertilization, that is called the double fertilization, and how these microspores and the megaspores are formed. This already explained in my channel, so you just check out. And this is the next stages of the plant development, that is the embryogenesis. Okay, so here the basic body plan of the sporophyte is established during the embryogenesis. So embryogenesis is what? Embryogenesis is a process or stage where the sporophyte's body is established or formed. And the two basic developmental patterns are seen here. One is the axial patterning. This is the axial patterning. And second one is that the radial patterning. Okay. Axial patterning means what? This is called the axial patterning. And radial patterning like this. This is called the radial patterning. Okay. So first this is called the axial patterning. And the radial patterning like that, the concentric ring-like structures formed surrounding the axes. Now, in the axial patterning, what will happen here? That the apical and the basal axes that is formed. Apical and basal axes formed. So, this is the zygote you can see here. And when the cell division occur, the two parts. This is the apical part and this is the basal part. The two parts, this cell divides to form the two parts, apical part and the basal part. So apical part, that means the shoot apical meristem or the upper portion of the plants, and the basal part that form the root apical meristem. So you can say in the short form SAM, that means the shoot apical meristem, and the root apical meristem, that is the RAM. Okay. So shoot apical meristem is at one end of the axis. So if this is the axis, the upper portion, this is here the shoot apical meristem that will develop, and this is the lower part, here the root apical meristem that will develop. Generally, two cotyledons are seen in the dicotyledonous plant, and one cotyledon is seen in the monocotyledonous plant. So the seedling or the embryo with one or two cotyledons are attached just below the shoot apical meristem. So cotyledons are generally attached one or two, that depending on the plant's types. But these are attached where? These are attached with the shoot, below the shoot apical meristem. Okay, below the shoot apical meristem. Clear? And there, so first the shoot apical meristem's development is seen, and then the root and the root apical meristem and the root cap that form in, from where? From the basal part of the axis. Okay. So this is the apical part that will develop to form the shoot apical meristem, and then the stem part, and this is the basal part, from where the root apical meristem and the root cap. Okay, so these are developed. And the radial patterning is what? It produces three concentric rings of the tissues. Generally, radial means I have told you that the ring the ring-like concentric ring-like cell development that will happening around or surrounding the axis. Clear? So three concentric rings are formed, or rings of tissues are formed. These are arranged with the radial axis, and the outermost layer, the three layers. So first the outermost layer that form the epidermis, and that covers a cylinder of the cortical tissue. This is called the cortex, and then which in turn covers the vascular cylinder. So if this is the axis, So here suppose three concentric ring-like structure develop here. This is okay. So outer one, that is known as the epidermis, and just behind this outer one, this is from the cortex region. Okay, and inside of that, this form the vascular cylinder. So this is the this form the this cell layer that form the vascular cylinder, this cell layer that form the cortex, and the outer cell layer this form the epidermis. Clear? So the two patterns are seen in the plant's development process. Now first one is the axial pattern. What will happen in the axial pattern? So here is example has taken, that is the Arabidopsis, Arabidopsis thaliana. This is the example, and here what will happen first? The embryo during the embryogenesis is established due to the first asymmetrical cell division of the zygote. So first zygote's cell division is occurred here in the embryogenesis process, and here giving rise an apical cell and a basal cell. So first this is the zygote and cell division occur, and the apical cell and basal cell that form. The apical cell undergoes to two longitudinal divisions. Next in the apical cell, here the cell divisions occurs in which way? In the longitudinal way. Okay, and this followed by a transverse division. That means first the longitudinal cell division is seen here, and then the cell division follow the patterns like the transverse path. Okay, transverse path. So longitudinal means the vertical way, and the transverse path, that means the horizontal way. Okay. So to form, how much cells are formed? The 8-cell octant stage globular embryo. So 8-cell octant stage globular embryo. This is the globular embryo that is formed, and larger vacuolated also divides horizontally, and here the vacuolated that cells divides horizontally at right angles to the axis. Clear? So first the globular embryo from the apical and basal, first the structure that is formed from the zygote, and then cell division occurred to form the early globular stage like structures. Okay, and this early globular stage that is formed, that is 8-cell octant stage globular embryo. First that is formed the 8-cell, it is known as the octant, and the globular embryo it is called at that moment. Okay, an early globular stage it is called. And then additional cells, or the cells, after divisions of 8 cells are formed. So then again cell divisions occur, and that cell divisions occur for what? To increase the number of cells. Okay, and the larger vacuolated also divides horizontally at. So what is happening here? The additional cells that divides to increase the number of cells of the globular staged cell. And now, generally the larger vacuolated also divides. One thing is missing, I think. So this is the basal cell. Okay, the larger vacuolated basal cell that divides horizontally at right angle to the axis. With the axis, these basal cells that divides horizontally. Okay, with the axis, this form a right angle and then horizontally they divides, and it gives a filament of 6 to 9 cells. And what is the result of this division? 6 to 9 cells that form, and which are known as the suspensor cells. Clear? So it gives the filament of a structure like that, filament like structure of 6 to 9 cells are there, and these cells are known as the suspensor cells. And that attached with what? This attached with the embryo. Okay, this attached with the embryo of the vascular system. And the basal cell that derivatives nearest to the embryo known as the hypophysis. So the basal cell that present nearest to the embryo, these are called the hypophysis. And here this divides, these hypophysis region cells that divide to form the quiescent center of root apical meristem and the root cap. That means due to the divisions of the cells that present that are known as the hypophysis, these divisions form the new cells and which form the quiescent center of the root apical meristem and the root cap. So first part of the axial pattern, how the globular stage formed, that is explained in this video, and this is too large topic, where how this root apical meristem form and the root cap form from the quiescent center. So these are also another process, and I will try to explain that topic in my next video. Okay, so you just continue with my channel, and this is the first part obviously of the embryogenesis. Clear? So thank you.